And we're going to move on to the next talk. So next talk is going to be given by Professor Ali Javey from UC Berkeley. So he obtained his PhD degree in chemistry from Stanford in 2005, followed by a postdoc experience at Harvard for 2 years, and then he joined the faculty of UC Berkeley. And he also is a senior faculty scientist at LBNL, as well as he serves as a program leader of electronic materials. So his research expertise is on integration of nanoscale electronic materials for various applications. And he's also recipient of numerous awards, to name a few: MRS Outstanding Young Investigator Award and Nano Letters Young Investigator Lectureship. So today he'll be talking about the wearables sweat sensors.
All right, can you hear me?
Yes.
All right, well, thank you very much again for inviting me. I think this is now my third or fourth time giving a talk at KAIST, and of course, previous times were all in person. And so I hope that in the near future I can once again give an in-person talk. So let me share my my screen. Okay, so I'm going to try to use the next 45 minutes or so to share with you some of our recent work on wearable sensors, and specifically wearable sensors for sweat analysis. And the big goal, the big vision is to move towards big data for human health. And we do believe that having non-invasive sensors, wearable sensors, could really provide you with a wealth of information about the body that you can use then for making all sorts of predictions about the body, as I will talk about. But that's really the long term goal, and I will share with you where we are today as well, of course. So let me start by giving you an overview of why we're interested in human sweat. I think, especially to Koreans, I don't need to tell you that the human sweat is very accessible. As you walk outside in summer, especially in Korea, you know that with high temperatures, the body can sweat quite a bit. And in fact, the human body can sweat up to 14 liters a day. I mean, just think about that number. That's really an astonishing number. Of course, that's under extreme conditions. Now, sweat is already known to be a rich source of information. There are many different biomarkers in sweat. And in fact, sweat analysis has been used in the past. This is off-body sweat analysis for a number of applications, including for doping for athletes, and also for medical applications, for example, for cystic fibrosis, or for example, for monitoring patients that have had a brain injury. These applications are already in use, but they're all done using off-body sensors. And in fact, the first mention of sweat in a medical context goes back to 1606. And there's a Spanish reference textbook, basically, that is listed here. And this is well before modern medicine. And here the reference was that if the baby has a salty skin, that baby tends to die young. And at the time, of course, it was not understood why that was the case. It was more or less just a myth. And today, it is believed that that reference, in fact, was to cystic fibrosis, and which I will talk about a little bit later. Now, in our lab, we are looking at these sweat biosensors for two very different applications. We are developing devices that can be used for fitness tracking. So we want to use these on athletes, on subjects that undergo prolonged periods of exercise, whether it's biking or running or other sport activities. We want to monitor, for example, how their biomarkers in the sweat is changing, the concentrations are changing, whether we can pick up signs of dehydration, whether we can pick up early signs that, for example, glucose levels are dropping. And the second type of applications is for medical or for health monitoring. So here we're talking about subjects that they may not be able to run for you. So these are subjects that you have to think about devices that can work with very small amount of sweat. Either you have to locally induce the sweating process somehow, or you just have to rely on natural sweating and somehow be able to analyze that very small sample volumes. And as I will share with you, we are always sweating, even when we don't notice it. If you're just sitting in a room at just the normal room temperature, you're sweating. And I will show you data on that. And the sweat rates often are very low, and the evaporation often takes place very fast, so you don't notice it. But in fact, that the small amount of sweat is enough for you to be able to collect and analyze it. And in fact, you can even measure that very small sweat rates, which I will show you without doing anything special. You just have to design devices with proper microfluidics, as I will show you. And then, of course, the goal is to be able to decode the composition of sweat. What can we tell from the changes in the composition of sweat? Can we pick up early signs of, for example, dehydration? Can we use that to predict certain onset of conditions? And then, of course, be able to use that for preventative care. And that's really the long-term vision moving forward. Now, as we started looking at sweat analysis several years ago, we very quickly identified a list of challenges, and over the years we have tried to address some of these challenges, and as we have done so, new challenges have showed up. So this is a dynamic list. And the very first challenge that we had to overcome was that you often deal with very low sweat rates, especially if you're looking at subjects that are in a stationary position. If you're just trying to look at the natural sweating events when the subject is at rest, you're talking about very small amount of sample volumes. And in analytical chemistry, you know that if you have very low analyte concentrations and low sample volumes, you're in trouble. So it's a challenge to be able to analyze those samples correctly and in a robust manner, such that you can trust the information that you obtain. Sample evaporation is a big deal. If your sweat rate is too low and you have evaporation taking place, then the concentration, of course, of analytes is going to get affected, and you may not be able to measure the concentrations correctly. So you have to design devices that can minimize evaporation. Contamination from skin is one that I want to highlight. This is early on, we were looking at glucose sensing, and we figured out that some of the hydrating soaps, they actually have sugars in them. And so if you're trying to measure very low concentrations of glucose that exists in sweat, but the subject had used some of those soaps that had sugar in them, then of course the question is, what are you measuring? And so one has to be extremely careful with basically making sure that there is no contamination from the skin. So when we do the experiments, we actually regulate what type of soaps the subjects use and lotions they use, and we have a protocol for cleaning the skin before mounting the sensors. And of course, being on the EE side, we have to make sure that the sensors have minimal drift. The sensor doesn't drift over time. If you just run the sensor with a constant concentration, the signal should not drift by much, but drift always exists. And so you just have to make sure that you can minimize it, and at the end of the day, the amount of drift that you get is going to be the error. The sensor error is going to be basically your sensitivity. Your sensitivity is not just determined by the lowest concentration that you get a response, but it's actually also determined by other factors, including by the sensor drift. And then finally, of course, we have to figure out what sweat composition is telling us. So we have to do subject studies and start to form correlations or look into the lack of correlations in some cases. So over the years, we have built an array of devices, and this is an image of one of the earlier devices that we made in our lab. From very early on, we looked at the problem from a system level perspective, and I am a material scientist and electrical and a device engineer. I don't do any circuits or systems. So this was an exercise for us to really start grabbing some of our devices and build a system around that. So here what you see is that the device has two components. There is the component that is shown as in white, the color is in white, and that's basically a very thin plastic substrate that has the sensors patterned on it, and these are these sensors, we call them transducers in electrical engineering terms. And this component is disposable. As I will show you, we make these using roll-to-roll printing processes. So we get a long sheet of them, we just cut them into small pieces, and they're effectively, it costs about a penny each. And they're integrated then with a electronic board. And the electronic board is what does, is in charge of doing the signal amplification, signal processing, conditioning, and also data transmission. So wirelessly transmit the data to a mobile device, for example, to a cell phone. And in electrical engineering, we refer the whole system as a sensor, and but often in other fields like chemistry, material science, you refer to the transducers as the sensors. So here we decided to take the full system approach, integrating off-the-shelf silicon chips with printed sensors for sweat sensing applications. Now, over the years, we have developed all sorts of different transducers for metabolites, for example, glucose lactate for different ions. Heavy metals are quite interesting. If you're exposed to heavy metals, the body tries to get rid of them right away, and you can pick them up very quickly, immediately in your urine or in your sweat. And this is well before they do show up in the blood tests, before you're poisoned effectively. And so in fact, it has already been shown by doing off-body analysis by other groups years ago that that you could do heavy metal analysis of sweat as a way of looking at early exposure signs to heavy metals. So we made wearable devices that can pick up immediate signs of heavy metal exposure. Many drugs do show up in sweat. Caffeine is the one that probably everybody is very familiar with. That shows up right away in sweat, and in fact, this is well known. For doping control for athletic applications, sweat analysis was used and has been used in the past because many drugs do show up in sweat, and it's very easy to collect sweat samples from athletes. And vitamins do show up. So we showed, for example, that vitamin C does show up in sweat, and in fact, the concentrations are correlated to the intake concentration. So you can start to measure the absorption rate, the metabolism rates of drugs, of vitamins, as you take vitamins. Skin temperature is informative by itself, but we also measure the skin temperature and also the sensor temperature as a way of calibrating the signal. Some of these enzymatic sensors are very sensitive to temperature. The enzyme activity depends on temperature, so you do have to do some calibration. And sweat rate is very informative, as I will show you. kind of to expand on this device a little bit more, this is our first generation device. Again, you can see the two components very clearly, and you can see that for the PCB, and in this case, the PCB, the printed circuit board, is flexible. So you can think about it as being part of the band of a watch, for example. It has 11 off-the-shelf silicon chips. So the 11 boxes highlighted here, each of them is one silicon chip, and but you can actually build just one chip that is custom built, an ASIC, and that does all of the jobs of all 11 chips. And in fact, one of my colleagues, Chris Pister, has already done that. So you can replace these 11 chips with one. We can do multiplex sensing, so you can put an array of sensors. In this particular example that I'm showing you, we had 4 different biomarker sensors, and we also had a temperature sensor. But in principle, you can pack many different sensors. The big question then becomes, how reliably can you fabricate X number of sensors with making sure that they don't drift and all that. And so we generally try to keep it right at this stage to a minimal number, because what we really care about is to get those correlations more than anything else. So we want to measure the correlations one analyte at a time. But in principle, of course, you can pack as many sensors as you wish. Just to kind of give you an overview of the sensor technologies that we're using, in principle, you can use different modalities of sensing for different analytes. You can, in fact, do also multimodal sensing just to get even more higher level of confidence in your data integrity. Most of the sensors that we have played around with in our lab are electrochemical sensors. These are based on well-established sensing technologies, commercially available. And so we just decided to use the best established sensor technologies, and we just basically transferred that to making these sensors on a plastic substrate in a reliable manner, such that the sensor-to-sensor variation is low, the drift is low, such that we can produce these at scale. And but the sensing mechanism is well established. And as an example, for the glucose and lactate sensors, we use enzymatic electrochemical sensors. So here the signal is current, and the current that you get from the sensor is proportional to the concentration of your glucose or lactate. And they're very selective. These enzymatic sensors are extremely selective. For ions, on the other hand, there are also electrochemical sensors, but the sensing modalities that we use is we use basically ion selective sensors. We put down membranes, iontophores that are selective to different ions. These are commercially available iontophores. You can buy them pretty much for most common ions. For example, for sodium, potassium, and other species as well. And what ends up happening is that the signal that you get is voltage, and the voltage depends on the concentration of the ion, and it follows the Nernst equation. And the Nernst equation is the same equation that we have in device physics, the very famous 60 millivolt per decade equation. It's actually the same physics. We just give it different names. And so here in this case, you get 60 millivolt of signal for 1 decade change of concentration, for 10x change in concentration. Same physics, again, same reason why it's 60 millivolt per decade. All right, and then for the electronic board, we have the different chips are shown here, but the key message here is that you really do need to have this electronic board if you do want to get your kind of have high confidence in the signal. The rule of thumb is that you really need to amplify your signal immediately, as close as possible to transducers. And that's really the key. The further you are from the transducers, the less your signal integrity is going to be. Now, for sweat rate measurements, the approach that we have taken is to basically design a microfluidic, and the microfluidic has inside of the microfluidic, we place our electrochemical sensor, so we can have sodium, potassium, glucose, or whatever sensor you want inside the microfluidic. The microfluidic has an inlet. The inlet sits on the skin. And in this case, the inlet is right in the middle. And that's where the sweat enters the microfluidic. And then along the channel, we place electrodes. And there are different ways to do it. We have different strategies for doing it. And you basically look at the impedance change as sweat goes through the channel. And by looking at the impedance of the electrodes, you can tell where the liquid front is in the microfluidic, and from that you can then extract your sweat rate. Now, putting a microfluidic ended up being really a key innovation because if you don't have microfluidic, your evaporation is huge. The evaporation loss is going to be very large. And so it's really important to have a microfluidic to minimize evaporation. We don't see any measurable evaporation with microfluidic, and you can convince yourself of that. And the reason for it is because the surface area of the liquid that is exposed to air ends up being just a cross section of the microfluidic, and that's extremely small compared to the total volume of the liquid that you have. So evaporation becomes extremely, extremely small. The other advantage is that we can avoid mixing of sweat as we measure the sweat. We can push the old sweat through the channel and keep measuring the fresh sweat. So if you're trying to do time resolved measurements, this is also more informative. Of course, you have to deal with diffusion. There's always going to be diffusion and back diffusion. And so we can model that, and we have done that, and you can correct for it accordingly. Now, in terms of the packaging, you can really explore all sorts of different device packaging, and here I'm just showing you one example packaging that we looked at several years ago. And we have different form factors. We're making devices that are in the form of a Band-Aid, and in some cases they're in the form of a watch. And this particular design is a watch-like device, so there's a display. And what the students did was that they implemented a flexible photovoltaic cell, and this is just an amorphous silicon cell for absorbing ambient light. And there is a flexible battery integrated, and that is being constantly charged by the photovoltaic cell. So think about this as your cheap calculators that have the photovoltaic cell that just basically run the device by ambient lighting. There is battery there so that in case you want to run these at night, the battery can do that when there is no ambient lighting. And so this I thought this was a clever application for the case of a watch band. This is really a perfect place to put a flexible photovoltaic cell, and you always have to have a band, and you can actually come up with clever designs and form factors for integrating that photovoltaic cell. And as long as your device doesn't need much power, and especially for these sensors that we're making, the amount of power we consume is quite low. So it makes perfect sense to take advantage of that. Now, this is an example set of data that we obtain. Here, I'm just showing you data from one set of experiments. On the top, I'm showing you the power. In this case, the subject is biking, so the power is the biking power. And so you can see that we gradually stepped it up, and then we kept the load, the power, biking power constant for an extended period of time. And then there was a cool down period where we stepped it down. At the same time, you can measure various physical parameters. You can measure, for example, heart rate, VO2, VE. You can measure skin temperature, lactate, glucose, sodium, potassium. So this is just one example set of data from one experiment. And you can imagine now we have sweat, or more recently we have sweat rate as well. We have all sorts of different analytes as well. So you can really kind of envision what I mean by big data. You can really collect a large set of data, and more importantly, be able to get dynamic data, time-dependent measurements, and look at how different biomarkers are changing with time. And I think those dynamic changes are probably more meaningful for sweat analysis. You can't do that today with blood. When you do blood testing, you're doing discrete measurements, and so you can't really capture those dynamic changes. And we think we should be able to capture those dynamic changes, and we are in fact doing that for some of the biomarkers by using sweat analysis. Now, for extending the platform for medical applications, it was very clear to us from the very beginning that just relying on exercise sweat is not going to do it, because for many cases you can't get the subjects to run for you. I was mentioning this to a cardiologist, and the cardiologist reminded me that for their heart patients, they actually get the subjects to get on the treadmill and to exercise for them for some time. But in general, it's not practical to do so, especially if you're trying to also get dynamic changes over extended period of time. And so over the years, we have looked at different platforms. The one platform that we looked at is to use a process called iontophoresis as a way of locally inducing sweating. And the idea for iontophoresis is that you have a hydrogel loaded with a drug, and you pass current through it. The drug goes into the skin. It activates the sweat glands locally. There are multiple FDA-approved drugs for this iontophoresis process. It is used by clinicians today for detecting cystic fibrosis. They even do it on infants, in fact. I will talk about a little bit later. So we basically use the same drug and the same concept, but the innovation here was that we made a wearable device that does iontophoresis and sweat analysis using the same patch. So in the middle here, we place our electrochemical sensors, and then we can measure different biomarkers using the same device. Now, more recently, we have developed a device that can measure at-rest sweat, your natural sweating events, without any induction. So the case of iontophoresis, we were locally inducing sweating using a drug. And in this case now, we are looking at natural sweat. We are not inducing it locally. And again, I want to highlight this, that we are always sweating. And as we are sitting or sleeping, we are sweating. And the sweat rates can be extremely low, but still there are finite. And so here the key was to develop a microfluidic device with proper dimensions, but then also the inlet, we had to figure out how to collect very small samples and then be able to actually push them to this through this microfluidic channel. So we came up with strategies for that that is highlighted here, but you can read the details in our paper. And so the devices now enable us to measure the sweat rate and sweat composition of subjects while they're at rest. And these are just some of the optical images of the devices as the students place them on the body. And in this case, we placed a dye, just a coloring dye, inside the microfluidic channel. That way, as the sweat enters the channel, there's a color, so you can easily see it. The color here is green. And here the patches were placed on 8 different locations on the body, highlighted by the positions on the left. And you can clearly see that the position 6, which is your fingertip, has the highest sweat rate. And so you can see that after 58 minutes, the fluid had actually gone through most of the length of the channel. So that's just in 1 hour. And so different spots on the body have different sweat rates, and we can actually measure them now. And in this case, the subject was just sitting at rest in the lab at ambient temperature. The temperature and humidity, relative humidity, are shown on the right. Now, in terms of doing subject studies, we really need to have access to a large number of sensors. We want to make sure that the sensor to sensor variation is also very low. And so for that, we started from several years ago, we started working with our colleagues, initially Professor Cho, who is now at SKKU here in Korea, or close to KAIST. Actually close to Seoul technically, and so we looked at different fabrication scheme, and we looked at, for example, doing roll-to-roll processing of these electrochemical sensors using some of their facilities, and there were some challenges with formulating the inks properly, and nevertheless, this is the end result. So you get a long roll of these plastics. Some of the rolls that we got is 100 meters in length. That's the length of the roll. And on this 100-meter roll, of course, you have thousands and thousands of devices. And so then we just have to cut them, and you can then test them. One of the worries that I had was that during printing, because you're using an ink, I was worried that the device-to-device variability would be large. The samples that we had worked with in our lab were made by photolithography and evaporation, so the metal quality was pretty pure, and lithography is a very well-established process. And to our surprise, the variation ended up actually decreasing. And so here I'm showing you the results from I guess close to 30 or 40 devices, maybe even 50 devices. And we tested them under different conditions. First, we just tested the bare electrodes, and we just coated them with a reference or a standard molecule, and we did CV measurements to just look up the reduction and oxidation characteristic of the molecules, and you can see that these different CV curves that are obtained from the different electrodes, printed electrodes, they're all pretty consistent. The potentials are very consistent. Both the currents and voltages are pretty consistent. We also functionalized these electrodes for different sensors. Here I'm showing you the functionalized pH sensors, and you can see that from the histograms that the distribution of the sensors in terms of their response, here I'm highlighting sensitivity, is pretty narrow. And again, this was to our surprise. I was not expecting this. I was expecting a larger variation, but it actually, the variation ended up narrowing. More recently, we have partnered with VTT, and this is an institute in Finland, and they are effectively one step below a manufacturing house. They have their tools are almost at the manufacturing level, just maybe one step lower, and they now do the roll-to-roll printing of the sensors for us, and they also do the roll-to-roll printing of or roll-to-roll processing of the microfluidics as well. So now we're getting roll-to-roll process sensor arrays with microfluidics, and the variation in this case is of course even lower, just because we are now using much better and more controlled facilities. Now, for the remainder of the time, so far I've talked about the technology. I'm going to use the remainder of the time to highlight some of the case studies and what we have learned by doing subject studies. What can sweat sensors tell us? I'm going to start with the work that we did early on, going to Stanford Medical School, to the Cystic Fibrosis Center at Stanford. And I mentioned to you already that the gold standard for detecting cystic fibrosis today, and that has been the case for the past several decades, is to do sweat analysis. And specifically, you look at sweat chloride ion concentration. And as a rule of thumb, is if the sweat chloride ion concentration is less than 40 millimolar, CF, cystic fibrosis, is unlikely. And if chloride ion concentration is more than 60 millimolar, then CF is very likely. And at that stage, CF is diagnosed. Given how cheap genetic tests are today, at this stage, then typically the clinicians order genetic tests. But in the old days, that was not being performed. In the old days, really, sweat chloride concentration was the only measurement that was done to diagnose cystic fibrosis patients. And it's a genetic disorder, and patients often die young, unfortunately. And so again, this was the myth that I told you before, that if you leak the skin of a baby and the skin is salty, the baby die young. They die young, and we think again that reference was for cystic fibrosis. And so here are some of the results. We made these patches that can do iontophoresis, so we locally induce sweating events. And the iontophoresis was done at minute 0, and so this is 20 minutes after iontophoresis. It takes some time after the drug goes into the skin for the sweat glands to get activated. And then you can see for a healthy subject, we were able to measure chloride and sodium ion concentrations, and in this case that I'm showing you, the concentrations of both are less than 40 millimolar. For the patient, you can clearly see that the concentration of chloride ion is quite high. In this case, it was over 90 millimolar. And what's also interesting is that in the case of patients, the cystic fibrosis patients versus healthy subjects, the concentration of chloride to sodium is reversed in terms of which one is larger. You can see for patients, chloride ion concentration is larger than sodium. For healthy subjects, they're closer to each other. Usually chloride is a little bit lower. And indeed, we were able to actually also detect that. And so we looked at multiple subjects, multiple patients, and the nurses helped us install these sensors on the patients. And of course, healthy subjects, they are easy to come up with. And in all cases, we were able to very clearly differentiate the healthy subjects versus the patients. And this was really a good sanity check, just because we went to a case where there is already sweat analysis being performed, but the sweat analysis, the way they do it is that they do iontophoresis, they wait until sweating's done, then they put a device to collect that sweat, then they send it to an analytical lab, the pathology lab, and then they get the results a few days later. Here you can get the results instantaneously, and in principle, you can do it at home. Another case study that we did was just looking at sweat rate, and we looked at sweat rate at different parts of the body, forehead, forearm, underarm, back, and we got the subjects to exercise for extended period of times, and we made correlations between the whole body fluid loss rate. So this is obtained by measuring the weight loss that you get from the subject when they're exercising, and that weight loss is simply from sweating events, is your body fluid loss, to the regional sweat rates. And you can see that especially for forearm and forehead, we were able to see a good correlation between measuring the sweat rate locally versus measuring the whole body weight loss or fluid loss. And so in principle, you can have these devices installed on the body and measure the sweat rate at just one discrete spot, and from that predict the body fluid loss. as a reference point, if you lose 2% body weight during exercise, that's one definition, commonly used definition for dehydration. So in principle, we can predict early signs. Now, I say in principle because there are some challenges here. Specifically, what we have seen is that while for the same subject, we do see these strong correlations, different subjects, they have different sweat profiles on their body. So for different subjects, some subjects, may sweat a lot more on their back versus forearm. So the calibration curve is actually different for different subjects, which does make it a little bit more complicated. Another interesting result is using our most recent sensors. These are the sensors that I mentioned to you, where we designed the microfluidic properly with the inlets properly, such that we can now measure the natural sweating events even when the subject is at rest without any induction. And so here the students did something very simple, which is just measuring the sweat rate. And they also measure the heart rate. On the top is the ambient temperature, just the room temperature. And they went around undergoing through different activities for one day. And so you can see that the subject at certain time points was eating, walking, sleeping, or just sitting stationary. And there is also a part that says Zoom talk. And so this is during the pandemic. This was a Zoom meeting. What's amazing is that you can see that during the Zoom meeting, the heart rate of the subject went up, but not by much. It went up, but not by a whole lot. And but what's amazing is that the sweat rate went up by 2 orders of magnitude, and this is simply because of stress. And when you give a talk, you probably have noticed that you sweat more. Now, we would have not been able to make this correlation directly if the talk was not a Zoom talk, because if you're standing up at the podium and moving around and moving your arm, then of course there is some physical activity involved. And so you don't know if giving a talk in person is usually an exercise process by itself, depending on how dynamic of a speaker you are. But in the case of a Zoom talk, you're just sitting stationary and you're just basically, your body is not moving much. And this changes all from stress. Another interesting experiment that we did was that one of the students installed these devices on themselves during a Zoom exam. And this was, again, during the pandemic. And they measured the sweat rate before the exam. So they obtained the baseline. And then during the exam, again, the sweat rate shot up by 2 orders of magnitude. And what was amazing is that it took half an hour after the exam was over before the sweat rate went back to the steady state. So you can use these devices as a mean of looking at the stress levels. And of course, if you just look at sweat rate by itself, that's not informative. You have to look at different parameters, and you have to know whether the subject, for example, is moving or not, and what's the ambient temperature. But if you have various information, you can predict signs of stress quite readily. So you can think about various applications there. Another interesting case study was with the case of diabetic subjects. And one of the diabetic subjects that we're working with told us that it was very clear to them that when their glucose level was off, either very high or very low, their sweat rate was dramatically affected. And specifically, what they had noticed was that when the glucose level is too high, their sweat rate goes down. And on the other hand, when the glucose levels are too low, the sweat rates shoot up. And in fact, in literature, this is a well-known phenomenon. And so we did some subject studies with diabetic patients. And again, we measure the heart rate. We measure SR. SR is sweat rate. And then we measure their glucose level. In this case, the glucose level is measured using a commercial device. This is a interstitial fluid sensor, a commercial prescribed sensor that the patient was wearing. And you can clearly see again that when the concentration of glucose is very high, the sweat rate is very low. And as they injected insulin, then the sweat rate shot up. And during the whole measurement, the subject was stationary. So you get a very interesting, you can pick up very interesting signs by just looking at natural sweat rate if you don't induce your sweating event on purpose. All right, so I just have a few more slides, and the final slide that I have is on drug analysis, and we think that that's really an interesting area to be able to, especially to study the metabolism rates for many medical conditions, especially the ones affecting the brain, and also for cancer treatment, for chemotherapy. The optimal dose range is pretty narrow. But the problem is when you take a drug, different subjects, of course, have different absorption rates. They have different metabolism rate. Those rates change depending on what activities you do, what food you take. So it's dynamically changing. And so keeping the subject in the optimal range is actually quite difficult. And so if you had the ability to measure those and kind of get a sense of the concentration of the drugs in the body, in principle, you can then have a feedback loop and kind of really enable personalized medication. And so the first case that we looked at is caffeine, and that's the easiest one to look at, just taking coffee. And this was done before we had our natural sweat sensors. So in this case, we had to get the subjects to bike for us. And so the subjects in this case took the coffee, took their caffeine at time 0. And then we measured the concentration of sweat caffeine with time. And what we saw is that initially the caffeine concentration went up, and then eventually the caffeine concentration decayed. And what you measure is that the maximum concentration that you get of caffeine, so it happens at time 60 minutes, about 1 hour after you take your coffee. So I always tell people that if you're giving a talk, the optimal time to take the coffee is probably half an hour before the talk, depending on how long the talk is and when the climax of the talk is supposed to be. And caffeine is an interesting one because it's also known to be it's already well established that caffeine sweat concentration is correlated to caffeine intake. People have done this measurements off body many years ago. Another drug that we have looked at is levodopa, and this is a drug that is used for Parkinson's disease. And so again, the same type of experiments. And again, we were able to measure the absorption rate and metabolism rate. And these just kind of give you a rough ideas of the type of things that we can do using these wearable devices. All right, so given the time constraints, I'm gonna stop here, and I'm just gonna conclude by listing some of the outstanding challenges. In particular, I wanna highlight the last one. I think it's all about decoding sweat. We really need to now do a lot more population studies, and that's what we're doing. We have done small scale population studies. We really want to do medium and large scale population studies. And as you make these sensors, really the key is to make sure that you can trust the sensor output. And that's the key if you want to make conclusions about sweat analysis. Finally, I have to acknowledge the most important people. These are the people that actually did the work. This is a picture of my lab from before the pandemic, and the people highlighted in bold were the students and postdocs over the years who contributed to the projects that I listed here. So with that, thank you very much. And I will take any questions if there are any, and if there's time available. I cannot hear you.
Hello?
Yes.
Okay. Yeah. Thank you very much for your wonderful talk. Your talk actually motivates me to go to the gym tonight and then make myself sweat, even though I don't have your sensors. So we have a lot of questions, but for the interest of time, I'm gonna just ask you 2 questions from the audience, and then the rest of them, I'll ask them to email you if that's okay, and you can respond to them with the email writing. So the first question is, when the subject is being studied, is the person able to drink water, or was it not permitted?
We have all sorts of experiments, and for all of these experiments, we have to have IRBs approved. So we have to write protocols, and we have to submit it to campus. There's a campus committee. When you do human subject studies, everything is well regulated. So it depends on what experiments we're doing. So if you're trying to measure, for example, the correlate the sweat rate with the body fluid loss, there we do not we ask the subjects not to take water and during the exercise. We have some other experiments where while we are doing the measurement, we ask the subjects to take certain drinks or certain food or certain drugs. So it just depends on the certain set of experiments. In all cases, we have IRBs, approved IRBs that we follow, the protocols that we follow.
Okay, thank you. And the second question is, if it is raining or really humid, then perhaps the sweat samples were contaminated?
Yeah, good question. So for the case of microfluidics, the skin, the part that we are collecting the sweat from, that part is sealed effectively, and so that's another nice advantage of having a microfluidic. Now, how does sweat rate get affected by humidity? And that's something that we are looking at. There are some information on that already in literature, but what we are doing is that some of our devices also have humidity sensors as part of the device, actually as part of the system. So we use commercial humidity, silicon chips. And so not only we measure temperature, but we also monitor humidity of the ambience.
Okay, thank you very much. So thanks again, your wonderful talk.
Thank you.
So let's thanks our speaker again.